Good morning and thank you for taking the time to listen to my presentation. This summer I will be studying pluripotent embryonic stem cells with Dr. Ihor Lamishka at the Black Family Stem Cell Institute at the Mount Sinai School of Medicine. So what are embryonic stem cells and why are they important? Well, an embryonic stem cell is formed from a zygote that's fertilized during in vitro fertilization. This zygote becomes a ball of cells, also known as a blastula. After 4 or 5 days, scientists remove the cells from the inner cell mass and place them in a petri dish where they begin to grow and divide. Growth factors are added to cause the cells to differentiate into nerve, bone, muscle, and pancreatic stem cells. In America, 17 people die daily awaiting a transplant, while 1,500 people die of cancer each day. Both of these diseases show promising signs of being treatable with stem cell research. Stem cells have the ability to differentiate into various cells that comprise the organs of the bodies. Doctors may eventually be able to utilize this ability to create synthetic tissues and organs, in turn saving lives and eliminating the need for a transplant or organ donor waitlist. Before we can begin to understand what work I'll be doing this summer, we need to understand the difference between the stem cells of the two basic categories, pluripotent and totipotent. A totipotent stem cell are stem cells that are the most versatile. When a sperm cell and an egg cell unite, they form a one-celled fertilized egg. The cell is totipotent, meaning it has the potential to give rise to any and all human cells, such as brain, liver, blood, or heart cells. It can even give rise to an entire functional organism. The first few cell divisions in embryonic development produce more totipotent cells. Unlike totipotent stem cells, pluripotent stem cells cannot give rise to an entire organism. On the 4th day of development, the embryo forms into two layers, an outer layer which will become the placenta, and an inner mass which will form the tissues of the developing human body. These inner cells, though they can form nearly any human tissue, cannot do so without the outer layer, so they are not totipotent but pluripotent. As these pluripotent stem cells continue to divide, they begin to specialize further.
Current studies at the Mount Sinai School of Medicine are involved in identifying the gene products of stem cells, sequencing the stem cells, and finding the factors that initiate differentiation in the stem cells.
The team led by Dr. Ihor Lamishka is currently looking at the genome-wide gain of function in the genetic screening of stem cells. Every cell in the human body has a complete template for creating an entire human. However, these cells will become very specialized and will not become an organism. So what the doctors and the researchers at the Mount Sinai School of Medicine are currently looking at is to determine how these cells differentiate. In 2005, Ihor Lamishka, Moshe Pritzker et al. Studied genome-wide gain-of-function genetic screen identifies functionally active genes in mouse embryonic stem cells. What they attempted to do was analyze the in vitro murine ES cells maintained in the presence of leukemia inhibitory factor, also known as LIF, which is a growth factor that activates certain genes. They wanted to determine which genes were causing these stem cells to differentiate. The team attempted to study these factors by submitting 11 genes to a microarray. These 11 genes were broken down into two subcategories, those that received LIH and those that did not receive LIH. A DNA microarray, also commonly known as a gene chip, DNA chip, or biochip, is a collection of microscopic DNA spots attached to a solid surface. Scientists use the DNA microarrays to measure the expression levels of a large number of genes simultaneously, or to genotype multitude regions of a genome. Each DNA spot contains picomoles, 10 to the negative 12 moles, of a specific DNA sequence, known as probes, or reporters or oligos. These can be a short section of a gene or other DNA element that are used to hybridize a cDNA or cRNA, also called antisense RNA sample, called target, under high stringency conditions. After comparing the two microarrays, the team found that AT1-PKB, CHOP10, CITED2, RHOJ, NC, and SPC1, PRUNE, and ASB6 were genes that were responsible for the rapid growth and specialization in the mice embryonic stem cells. The comparison of the LIF screen and the negative LIF screen confirmed their beliefs. In 2009, Dr. Ihor Lemishka's team attempted to revolutionize the study of gene expression. By utilizing GATE, a software which was developed for the analysis and visualization of high-dimensional time series, they created a series of code that would allow them to analyze the gene expression in cells. By using GATE's ability to differentiate colors, they would create a specific subset of code that would allow each color to represent a specific gene. When this color would pop up in the software, they would be able to understand which gene was being expressed, in turn affecting the phenotype of the given cell and the differentiation of the stem cell. In 2010, as a culmination of their previous studies, the team led by Dr. Ihor Lemishka studied SVM classifiers to predict genes important for self-renewal and pluripotency of mouse embryonic stem cells. They developed a classifier, which is a supervised machine learning framework for predicting self-renewal and pluripotency MESC stemness membership genes, using support vector machines. The data used to train the classifier was derived from MESC-related studies using mRNA microarrays. By using the SVMs to predict the genes, the scientists would be able to predict the outcome of the phenotype of the gene and therefore the differentiation of the stem cell. The application of SVM classifiers to predict stemness genes applied on test sets of the two independent genome-wide RNAi screens that identified candidate genes functional for self-renewal and insulin signaling were found to be fairly successful. The positive set of data based off of the ESRNAi screen was found to have a 50% and over success rate in predicting the phenotype of the genes. The work being conducted at the Mount Sinai School of Medicine and the Black Family Stem Cell Research Institute in conjunction with Dr. Ihor Lemishka is attempting to create a streamlined process for following the differentiation of stem cells. By creating a streamlined process, they hope to create an ability to specialize stem cells based upon need in order to create synthetic organs that can one day be made and placed inside humans to alleviate the need for a transplant list or a donor list and a waiting period for receiving such organs. A synthetic windpipe such as being shown is the first synthetically made stem cell product. However, it took months of designing and specialized cells. If there is an ability to program the cells to create these, much would be saved. Thank you for giving me your time and your attention. At the conclusion of this bibliography, I will answer any questions that you may have.